Welcome to Trial Site News Weekly Roundup. Today, we'll be talking about the U.S. Army Medical Research and Development, which has developed a COVID-19 vaccine candidate, SpFN, which is now in clinical trials. Followed by a story on RLF-100 expanded access program at the Houston Methodist Hospital, ninefold, which appears to increase probability of survival from critical COVID-19. Then we'll discuss Math Plus protocol, which includes ivermectin option for prophylaxis and early treatment of COVID-19. From Trial Site News, I'm Adrian, and the Weekly Roundup starts now. So, for our headline story of the week, U.S. Army Medical Research and Development announced the commencement of Phase 1 clinical trial soon for a novel vaccine designed to take on SARS-CoV-2, which of course is the virus behind the pandemic. That was first delivered via a press release on October 14th. The director of Emerging Infectious Diseases at the USAMRDC's Walter Reed Army Institute of Research reports encouragement about this particular approach for this internally developed vaccine. It's officially called spike ferritin nanoparticle, or SpFN. The vaccine candidate employs ferritin, a protein found in almost all living organisms. Now, with this input to help achieve the intended purpose, the scientists seek to block the COVID-19 infections by attaching a certain type of spike protein to a polymerized version of ferritin. Now, testing has already commenced, at least from a preclinical research basis. The director commented on the process of developing this vaccine candidate and said that the results we're seeing and the methods that are being applied to our vaccine in the animal studies are showing consistent results from the methodology so that we can benchmark it against the other vaccine candidates. And so, the Army News press release shared that the recent announcement was made at an annual conference, virtual this year of course, called the Association of the United States Army, or the AUSA, event. Speakers included Lieutenant General R. Scott Dingell, U.S. Army Surgeon General and Commanding General of the U.S. Army Medical Command, or AMC, and Mr. John Resta, Director of the U.S. Army Public Health Center. Now, during the pandemic, U.S. Army scientists commenced the ongoing development of several vaccine prototype candidates capitalizing on a platform previously tested in January. There has been a fast and furious attempt to identify the vaccine strain with the strongest immune response to the SARS-CoV-2 virus. Now, once potential candidates were identified, they were sent to the U.S. Army Institute of Infectious Diseases for large animal efficacy testing. And during the press briefing, the Army and other Department of Defense subject matter experts were mobilized to offer and advise and counsel to other Operation Warp Speed vaccine development efforts. Now, Trial Site News will track any public updates with SpFN, while dedicated scientists working under USARMDC pursue candidates for future coronaviruses. Patients with critical COVID-19 and respiratory failure treated with RLF-100 or Aviptadil have a ninefold increased probability of survival and recovery from respiratory failure than those patients only on the standard of care, reports Relief Therapeutics Holding AG and NeuroRx Incorporated, based on a recent summary of expanded access protocol results from Houston Methodist Hospital. Now, thanks to the heroic work of Dr. Youssef and team there in Houston, as well as in this investigational drug, lives have been saved. A separate randomized controlled Phase 2B and 3 clinical trial, which is to be completed by November, should offer more evidence whether RLF-100 exhibits the definitive results for FDA approval or emergency use authorization. Now, COVID-19 related death is primarily caused by respiratory failure. Before this acute phase, however, there is evidence of early viral infection of the alveolar type 2 cells. These cells are known to have angiotensin-converting enzyme 2, ACE2 receptors at high levels, which serve as the route of entry for the SARS-CoV-2 into the cells. Now, coronaviruses are shown to replicate in alveolar type 2 cells, but not in the more numerous type 1 cells. These same type 2 alveolar cells have high concentrations of VIP receptors on their cell surfaces, giving rise to the hypothesis that VIP could specifically protect those cells from injury. Injury to the type 2 alveolar cells is an increasingly plausible mechanism of COVID-19 disease progression. Now, these specialized cells replenish the more common type 1 cells that line the lungs. More importantly, type 2 cells manufacture surfactant that coats the lungs and are essential for oxygen exchange. Now, other than RLF-100, no currently proposed treatments for COVID-19 specifically target these vulnerable type 2 cells. Now, in an open-label prospective study involving 45 patients, 21 of them were admitted to an intensive care unit with critical COVID-19 and respiratory failure and treated with RLF-100, as compared to 24 control patients treated in the same way. All patients had severe comorbidities that rendered them ineligible for the ongoing randomized controlled phase 2b/3 trial being conducted to ascertain safety and efficacy of RLF-100. And all patients were deteriorating despite treatment with approved therapies for COVID-19. Now, this study, NCTO 445-3839, was part of the Expanded Access study. Now, overall, 81% of RLF-100 treated patients survived beyond 60 days compared to 17% of control patients. Patients treated with RLF-100 demonstrated a 9-fold increased probability of survival and recovery from respiratory failure with a high degree of statistical significance. The statistical analysis for this study was performed by Professor Phil Levin. Now, Dr. Youssef led the study. The section chief of general academic pulmonary medicine at the Houston Methodist Hospital reported that we are encouraged by these initial results in highly comorbid patients with COVID-19 respiratory failure, and we are pleased that the majority of these patients have returned safely to their families. We look forward to the upcoming results of the randomized double-blind prospective trial in less severely comorbid patients for confirmation of these results. Now, NeuroRx Inc. maintains the U.S. intellectual property rights to RLF-100, licensed from Relief Therapeutics. Their CEO, Dr. Jonathan Javitt, reported that we are grateful to Dr. Youssef and to the Houston Methodist Hospital for having the courage to treat and study patients at this level of risk. The results suggest that there may be substantial hope to mitigate the attack of the coronavirus on the delicate cells that line the lung with a natural peptide that has been protecting the lung's lining since humans first walked the earth. Now, while the number of patients treated at Houston Methodist is modest, the initial results in our nationwide expanded access program suggest similarly encouraging survival with RLF-100. We will continue to monitor treatment with RLF-100 in other hospitals. Now, vasoactive intestinal polypeptide, or VIP, was first discovered by the late Professor Sami Said in 1970. Although first identified in the intestinal tract, VIP is now known to be produced throughout the body and to be primarily concentrated in the lungs. VIP has been shown in more than 100 peer-reviewed studies to be a potent anti-inflammatory anti-cytokine activity in animal models of respiratory distress, acute lung injury, and inflammation. Most importantly, 70% of the VIP in the body is bound to a rare cell in the lung, the alveolar type cell 2, that is critical to transmission of oxygen to the body. VIP has a 20-year history of safe use in humans in multiple human trials for sarcoidosis, pulmonary fibrosis, asthma, allergy, and pulmonary hypertension. Now, Trial Site News will continue to monitor the Phase 2b/3 clinical trials for updates. The study, NCTO 4360096, concludes this November. You can sign up for Trial Site News newsletter for updates. So Math Plus is the COVID-19 protocol by Dr. Paul Marik and the Frontline COVID-19 Critical Care Alliance, or the FLCCC, as we previously reported here at Trial Site News. And while the NIH is still recommending ivermectin be only used in clinical trials, the September 28th update to Math Plus includes this drug as described below. The protocol noted that it is our recommended approach to COVID-19 based on the best and most recent literature. This is a very dynamic situation. Therefore, we will be updating the guideline as new information observes. So, beginning with prophylaxis or prevention, the protocol notes that a safe and effective cocktail of substances may prevent or mitigate COVID-19. For this purpose, Marik recommends melatonin, high-dose vitamin D3, the deficiency of which has been tied to increased risk of catching COVID-19 and worse outcomes, vitamin C, zinc, and famotidine. For post-exposure prophylaxis, home treatment during the acute phase in mildly symptomatic hospital patients, ivermectin is listed as an optional medication. The Math Plus protocol emphasizes that COVID-19 has different phases requiring different approaches. For example, antivirals likely only help during the initial viral replication phase, and anti-inflammatories are expected to be effective during the pulmonary phase and possibly the post-COVID-19 phase. And as patients progress down the pulmonary cascade, the disease is much more difficult to reverse. Now, to quote, the early treatment in the pulmonary phase is essential to a positive outcome. In this phase, Math Plus indicates anti-inflammatories for cytokine storms plus anticoagulants to limit vascular clotting. Now, if the storm is not stopped, often patients do not recover. The scientific basis for Math Plus protocol is reviewed in an article in the Expert Review of Anti-Infective Therapy. And an interesting talk from Dr. Merrick on COVID-19 can be found there. So, who is Dr. Merrick? Well, the doctor is Chief of Pulmonary and Critical Care Medicine at Eastern Virginia Medical School in Norfolk, Virginia. His work also includes over 400 peer-reviewed articles, 50 book chapters, and 4 books on critical care.